Katalina Eriksson. I'm a bioinformatics programmer for the Scripps Translational Science Institute, and I'll be talking today about the Scripps Genome Advisor. Adviser is a solution for whole genome annotation and prediction, and it annotates based on genomic elements, genes, transcription factor binding sites, microRNA binding sites, etc. And depending on the genomic element that is impacted by a variant, we attempt to make a prediction at whether or not the genomic element is functionally perturbed. We make a prediction for just about every way a gene can dysfunction. Today I will talk about the real case study, idiom study, which is idiopathic diseases in man happening here at Scripps, and example based on patient number 9, which is a 9-year-old European female, lifelong history of hypotonia, developmental delays, cognitive impairment, intermittent agitation. MRI showed subtle asymmetric volume loss in left hippocampus. Motor milestones were delayed. Onset of generalized tonic-clonic seizures was at 4.75 years of age. And we did quartet exome sequencing, and the exome sequencing variant summary, we had 101,797 SNPs and intels, 18,475 rare variants at 1% allele frequency, 1,053 nice anonymous truncating or splice site variants, 2 de novo inheritance variants, and 3 homozygous or compound heterozygous variants. And I will show you how we found the variant that we think caused disease. So we had the exome sequencing variant file. To upload the file, you go on genomics.scripps.edu, and you can upload the file for annotation. You can register to create an account or log in. We also accept unregistered uploads. I have an account. I log in. You can see the variant file format that we accept, and it's VCF, complete genomics, regular tab delimited format. To upload file, go here, choose file to upload, desktop, and I will upload the VCF file. Depending on the file size, that might take a few seconds, but I already have to upload, so this file is quite big. I did already annotate this file, and you go on download results, and I already annotated it, so I download the summary file. You will get an email once the file is annotated, and it can take from 10-20 minutes, sometimes maybe up to an hour, depending on the file size. To visualize the file is quite big, so it won't fit into Excel. You can use Adviser UI. You can download it from here. So it's a desktop applications for Unix, Mac, Windows computers, and you can download the file of the UI. And downloads, and start the application. You use the command for Unix and Mac, and the bat file for Windows computers. So you start the UI, file, open, desktop, 9. You upload the summary file, and you have a progress bar that tells you how long it's going to take. And this is how the summary file looks like. You have gene type, non-coding, protein coding, location, intron, downstream. You have conserved elements, allele frequency for 1,000 genomes, Welderly allele frequency, UQTL, microRNA. In total, it's 85 annotations. GBSNP, PharmKHB, and probably one of the most interesting will be Adviser score, which is a category 1 to 5 based on ACMG categorization. So for example, we have sort functionality, we have 13 custom filter saved file once you have a reduced set of variants that you decide to save, summary statistics, help. So I'll show you a few examples of custom filters. So for example, non-disease filters variants category 1 to 5. And you have a few category 1. Filters only category 1, so you have a category 1 variants here. Since this is a quartet, so you don't know if the impacted patient has the variant, you can load the genotypes, compare load VCF file. Film 9. So all of the genotypes are loaded. This is the entire file. Once again, you do the custom filter. And when you look here, you see right here is the proband. This is mother, father, mother, and the sister. None of those variants are de novo or homozygous. No, none of the category 1 variants. So this probably is not a good filter for this exact case. To move in between filters or to go to the original data set, you press show original data here. Some other filter will be chromosome position. So you filter depending on chromosome if you have any gene of interest. We have a simple filter, advanced filter, novel variants, cancer genes, and you can read about all of this filter on our website, the description on what exactly each filter does. So for this exactly, once you loaded the genotypes, one interesting filter will be the EDN filter, which will extract all de novo variants, homozygous, in child only, or compound heterozygous filters. So you press EDN, father is unaffected, mother is unaffected, proband is affected, and sister is unaffected. Filter. So you just see a handful of variants. We look here, none of them are categorized as advisor scores, so there is no prior knowledge about any of these variants. There are some homozygous, some compound heterozygous, and the variant after our conclusion, we came to conclusion that this variant on gene KSNB1, if you look in polyphen, right here, polyphen is probably damaging, and this is this variant wasn't known to be disease causative in the literature, but based on its function and other genes known to cause epileptic encephalopathy, it was the most likely candidate. We then found 2 other individuals that had epileptic encephalopathy, and they had deleterious variants on gene KSNB1. So this is a de novo variant in proband. And as I said, we found 2 other individuals, and the paper was recently published by Dr. Turkomani, my PI, de novo KSNB1 mutation in epileptic encephalopathy. Something else I will show you right before we leave. Once you have this reduced set of variants, you can save this to file for if you need test.txt. If you need to later get back at this file and open this reduced set of variants. And thank you to Ali Turkomani, Philip Pham, and Billy Shea.